So next experiment students we are going to study is that to test the glucose, sucrose, starch, protein and fats. The major organic nutrients which are substances which are required for the living organism for the growth and maintenance of the body organism. Especially we are going to study what type of nutrients we are finding out in the different plant body. So, as we know, the major nutrients required for the body is carbohydrate, which is the main source of energy for our body, made up of carbon, hydrogen, oxygen, and with the formula of CnH2nON. It is broadly being classified into three groups: monosaccharide, which is the simplest carbohydrate, which cannot be hydrolyzed. Second one is your oligosaccharide, that is formed by the few molecules of 1 to 10 molecules of monosaccharide units and by the glycosidic bond. That is like lactose, maltose, etc., which can be studied, which is basically sucrose is the non-reducing sugar. You might have studied under the theory, like in case of monosaccharide, which is having the reducing sugar, which can be tested by Petri solution or Pellic solution. Then third type is your polysaccharide mostly, which is being consumed as a food. There is a polymer of monosaccharides made up of several molecules of one or more types of monosaccharide like starch, cellulose and glycogen. Starch is the important constituent of the plant body and glycogen in case of animal body. Second part is your protein, which is made up of, this is the most important nutrient which is required for the living body, which helps in various ways for the growth, development of the living organisms, whether it is immunity, helps in digestion. Most of the hormones are made up of the proteins, so that you have read it in your theory. So, proteins, which are the complex organic compounds, which are made up of carbon, hydrogen, oxygen, nitrogen, phosphorus, or sulfur. So, here we are going to study about the constituent of the protein in the food material, and especially as I told you, it is in the plant body we are going to study. And the third type is your fat, which is another organic compound made up of carbon, hydrogen, and sometimes oxygen, which are nothing but the ester of fatty acid and glycerin, which are rich source of energy and mostly found in the oil seeds. And animal tissue, it is almost present, like in egg post tissues, the fats are present. So, what are the ways you are going to do? Mom will explain you about the method of conducting the test. So, just I am explaining you how to do it. After that, Mom will explain you. The first thing is that you will have to test for the carbohydrate. So, you have to take the material. Then it is a varied test which has been given here. I am not explaining more about it. Second is your Pellic test, which is also being done to study the presence of the glucose. Then test for sucrose, which is being done, the confirmatory test is being done by taking the magnetic solution. Next is your test for protein. Basically, three types of tests are done. One is your Leydig test, second one is your genetic test, or third one is your Magen's test. Either of these tests you can do to study the protein present in the food material. Third one is the presence of fat, which is being done where you are going to take the B node or the castor seed, where you are going to dip it in the, then after that you are going to add in it the sodium 3 to find it out, the presence of the fat. So now I will ask Mom to explain you how the tests are being done. So she will explain you in detail about it.
Okay, now I'm going to demonstrate the test for glucose. For glucose, you have to take plant source, that is ripe banana. So you have to mash ripe banana and take small quantity of ripe banana in a test tube. ripe banana towards the bottom of the test tube. Next, you have to add Benedict solution. This is Benedict solution. Take 4 to 5 drops of Benedict solution. Okay, color of the Benedict solution is blue. So, the glucose is a reducing sugar, so it generally reduces cupric to cuprous. That's why this blue color when heat will convert to brick red color. Look here. It is blue in color, Benedict solution. Now this Benedict solution react with glucose. You know, ripe banana contain fructose. Glucose and fructose are reducing sugar. So gradually this blue color turn to green. Just wait. You have to heat it. Keep this test tube in a slanting position. And don't keep
away from the bottom.
And don't keep constantly going. Just move out. Take it.
These are the precautions you people have to take care of it. Keep
And
away
look
from
at
the
this.
body.
Now green color turn to green and gradually turn to brick red color. Look here, color is changing. Now it is turned to brick red color that indicate presence of glucose in banana, right? Banana. So this is the test for glucose. Next, you will test for starch. Starch is a polysaccharide, it is a part of carbohydrate, it is a polysaccharide. So, it will not give any taste with Benedict, but give taste with iodine. This is the iodine solution. So, you have to take a test tube and add starch solution. Starch is the main plant source. You know, potato also rich with starch. Rice, wheat, all are rich with starch. So when you add iodine, it will turn to blue-black color. Actually, stir it, then add, give blue-black color. This is blue-black color that indicate starch always react with iodine and give blue-black color. Next is test for protein, and plant source of protein is peanut or almond. Okay, this is the rounded almond. So we have to take 5 milliliter of almond. This much, and you have to carry out bioweight test. Bioweight test means you have to add equal amount of copper sulfate and sodium hydroxide. This is sodium hydroxide, dilute sodium hydroxide. So look here, color gradually turn to purple color. That indicates presence of protein. So it will gradually convert to dark purple. After some time, to here, it will convert to this type of color, purple color. That indicates presence of protein. Next is test for fat. So various tests are there. You can dissolve this fat in benzene. But one most important test is test with Sudan 3. Sudan 3 is the reagent that is used to test for presence of fat. So I have taken oil, sunflower oil, and 2 to 3 drops of Sudan 3. Look at this type of color will form. The pink color bubbles formation occur that indicate presence of fat. So all these are tests for food source. That's all. Thank you.